A-level revision video on muscle contractions, and this is an updated version that I am doing for 2024. Okay, and this I've done this version because basically I looked back at the old version, I wasn't happy with it, and I think it fits the modern specification better than my previous video. So let's have a look at this then. What we're going to look at today is the structure of the sarcomere. We're then going to look at what the different patterns are that you can see in that picture there. So the A band, the H zone, all that sort of stuff. And then we're going to look at the key events in muscle contractions, which is probably the hardest question that you'll get on this. And then we're going to look at how the calcium ions are involved in muscle contraction control. A really important question, that one with the calcium ions, because if you got the essay question of the importance of ions in biological molecules or biological organisms in the paper 3, then you could answer that in detail. But let's get a move on, because otherwise we'll be here all day. Introduce you, we are focusing on something called striated muscle. It's also known as skeletal muscle, but under a microscope it looks striped, and therefore it has this name of striated muscle. And those stripes are key because they're the different proteins that are going to be working together to allow the muscle to contract. Basically, we might call the cells, and I might refer to it as muscle cells or muscle fibers. So because the cells are very different to other cells, they're often referred to as fibers. They're surrounded by a cell surface membrane called a sarcolemma, so you should be using that word rather than just a membrane. And they contain many mitochondria and specialized tubes or tubules that store calcium ions, which we'll come on to later. And instead of being called endoplasmic reticulum, although you probably get the mark for that, we should technically call these the sarcoplasmic reticulum. And basically muscle fibers are in bundles, okay, surrounded by connective tissue, and we call these therefore we'll refer to as muscle fibers during this video. They have a good blood supply of oxygen and glucose, obviously, hopefully, to allow aerobic respiration via mitochondria. And you'll see the need for ATP throughout the muscle contraction stages in a few minutes. What do we mean by a sarcomere? So sarcomere is one individual unit of the muscle fibre or the muscle fibril, and it spans the distance between two Z lines. So let's have a look at this and try and explain it in a bit more detail. So one sarcomere is the distance between these two Z lines here, and the Z line is at the end of the fin filament. So you see these fin filaments in orange here where they end, that is the Z line. Okay, the fin filament is a muscle called actin, which we'll be talking about a lot in a few minutes. And then in the middle of the sarcomere, there is this red bit here. There is the thick filaments, which is the protein myosin, and that's one with the heads on it that we'll be talking about in a few minutes. So there's a couple of different bands that you need to know. So the Z line is the end of each sarcomere. You've got the I band, which is obviously here, but also over here, and that is just where there is fin filament here. Okay, and then you have the A band, which is the region where both the thick and thin filaments overlap. And then there is also the H zone, which is where there is just thick filament, which would obviously only happen when the muscle is relaxed and not contracted. Now, those are terms you need to know. You need to be able to remember this picture, this image. So I think we need to get a bit more. Again, just to give you some different versions of it. So if we start at the bottom here, I think is probably the best thing. Okay, so here we have let's have a look at here. So we've got the Z lines here, we've got the thick filament here, and the thin filaments obviously over here. Okay, and remember we said the I band was where there was just thin filament. Okay, and this therefore is quite light on the if you were looking at it under a microscope. Where you get the A band, where there is crossing over, it's a lot darker, and when there's just, I suppose, the H zone, which is here and here, just myosin, then you get the A band. Okay, now why is this the case? Well, basically it's the case because of this. If you look at this, we've got A here. This is where actin and myosin are overlapping, so you get a darker colour. You get B here, which is the I band, which is just the thin filament, and then you get the A band, which is obviously got a bit of the darker colour of the myosin. So basically it's the amount of overlapping of the different proteins that causes the different colours in the sarcomere. Remember, the sarcomere is the distance between two Z lines in a muscle fibre. Old version of it, but I think quite a nice version of it again. So again, we've got the H zone. Excuse me, we've got the H zone, which is where you just have the thick filament, okay, which gives you this sort of colour. You've got the A band, which is the actin and myosin overlapping. You've then got the I line, which is where you just have the thin filament, the actin, and you've got your Z lines at the end of the sarcomere. So again, A bands are dark when stained, as both actin and myosin are present. The I band is where you only have actin present, so it's lighter, and the H band is sort of in the middle because you only have myosin present. So that's it. Now, the key thing that we're going to be looking at today is, well, how does this sarcomere overlap? And effectively, when a muscle contracts, what we are trying to do is what the muscle is doing is moving the thin filament, okay, this way inwards, and moving this thin filament, the actin, this way inwards, okay, and therefore you're reducing the distance between two Z lines during contraction. So the myosin is thick filament and it has head space in either side, which we'll talk about in the next few minutes. And then the actin is a globular protein, has two chains wrapped around each other, and we'll talk about that. So there's an image of the myosin, the thick filaments there with the heads, which are important. So I said to you a moment ago on the last slide, this is all about making this sarcomere decrease in size when we're talking about contractions. So the sarcomere contracts when the myosin heads form cross bridges with regular receptor sites on the actin, and they're spaced out. And then using ATP, don't think it's that simple though, we're going to talk about it in a few minutes, they move the actin towards the center of the sarcomere, making the actin and myosin overlap. So you can see here the distance between the Z lines is much greater when they are relaxed to when they are contracted. The distance between the Z lines becomes much less as a result. And the reason is not because of the thick filament moving. The thick filament sort of pulls along the thin filament, the actin, along. So you can see here the actin has moved from here and it moves inwards like that to the center of the sarcomere, reducing the distance of the sarcomere, making the two overlap more, and therefore giving it, I suppose, a bigger A band and a smaller H zone as a result. We look at how this actually works. The evidence for this is that the myofibrils appear darker when they overlap, and we can see this in contractions. As I just said a moment ago, the I band, which is the region where between the two Z lines becomes narrower, okay, because there is more overlapping between the two. The Z lines move closer together, shorten the sarcomere, and the H zone, where it's just in the middle, the thick filament, the myosin, also gets thinner as a result. So we can see this under a microscope during contractions of the muscle fibers. Structures are really key, and you need to know the difference between the two. You may see, if you look at older books, something an extra protein here called troponin, which I've actually blanked out here. You see it was on here. We don't need to know what troponin is for A-level biology. We just need to know about tropomyosin, which is an extra filament. So we've got two different main free proteins involved, I suppose. Myosin. Myosin is a Let me just get my thing up here. So myosin is the thick filament. It has heads on the end, okay, and you can see how they're aligned in filaments here. There's lots of them together with lots of heads. And then you have actin, the thin filament. That actin has an extra protein wrapped around it called tropomyosin. So you need to know from the top of your head, you need to be able to memorize the fact that the fibrous protein myosin forms a thick filament. It has two protein types, a fibrous protein arranged in the filament with lots of tails, and a globular protein with two bulbous structures making two heads at the end. The thin filament is made up of actin, and actin is a globular protein made of long chains twisted around each other. So you can see the blue chains at the bottom there. And there is something called tropomyosin, which is a long thin thread wrapped around the actin that blocks its binding sites when we're not contracting the muscle. Let's have a look then at how this works. So if you need a break, it'd be a good time to have one now, because looking at muscle contractions is a long, long job. So let's start off with what happens at rest. So when the sarcomere is resting, when it is not contracting, the actin has binding sites that are blocked. As you can see here, if you go to these little dimples in the actin are the binding sites. They are blocked by a molecule called tropomyosin, and that prevents the attachment of the thick filament of the actin, of the myosin heads, to the actin receptor site. And that is essential for to be removed if we want to cause the contraction of the muscle filament. So let's go through then what actually happens during muscle contraction. The muscle has to be stimulated. Now this will be covered probably in another lesson by your teacher, but let's have a look at this. This is called a neuromuscular junction, and I'll go through it relatively quickly, but it's important you can talk about the stages of this. So neuromuscular junction will be at the end of a motor neuron, because the motor neuron connects to the effector. Remember, an effector which causes a response is always a muscle which contracts or a gland which releases a hormone. So the motor neuron, the action potential rises down the end of the motor neuron. The action potential causes then vesicles to fuse with the presynaptic membrane, and that causes the neurotransmitter, which is acetylcholine, still same as in a cholinergic synapse, to diffuse across the synapse. This side, however, we then have, instead of a postsynaptic, instead of another neuron, we now have the muscle fiber membrane, or the sarcolemma. So the sarcolemma has receptors on it, just like in a synapse in normal regular postsynaptic membrane, and that the acetylcholine will bind to those receptors. That then causes depolarization, just as before, so sodium channels open, sodium rushes in, and that causes a wave of depolarization to travel along the membrane. But as the muscles are adapted to have something called T systems, and think, why are they called T systems? Well, if you just think about it logically, this looks like the letter T, okay? And the T systems, the impulse travels down the T systems, and that causes depolarization, which causes calcium ions to be released from sarcoplasmic reticulum, which is specialized endoplasmic reticulum. The calcium ions then bind to proteins in the muscles, and then that causes muscle contraction, which is what we're going to look at in a minute. So the whole muscle contraction is controlled by the availability of calcium 2+ ions. Now the next bit then is acetylcholine esterase. So just so you're aware of this, we're talking about it, that breaks down the acetylcholine that's gone into these receptors, and then that returns back to the presynaptic neuron using ATP. But obviously for this bit, we're not really interested in this section today. We're just interested in how this nerve contracts. And the sliding filament mechanism step by step. And remember, if you get asked to describe the actions in a muscle contraction, this would be what you'd be expected to talk about. They would more likely ask you about neuromuscular junctions if they wanted you to talk about the previous slide. However, if this came up as an essay, you could link it all to do with calcium ions and the importance of them. So step one then. So this is the slide, the image I showed you before. So we have the muscle at rest, the sarcomere at rest. So at this point, the myosin filament, its head is not able to bind to the actin filament. And the reason is that the actin filament has tropomyosin blocking the binding site. So it is unable to attach to the actin receptor site. Now notice before we move on though, there is one important thing I want to make sure you're aware of. There is ATP attached to the myosin head, and that, sorry, ADP I should say, and that ADP is important because without that the myosin head will not be able to attach to the actin. So let's move forward. So obviously from what we just talked about with the neuromuscular junction, calcium ions arrive, and the calcium ions cause the tropomyosin, which is the red bead-like string wrapped around the actin, to have a conformational change. So it changes the shape of the tropomyosin. Now that causes calcium ion, that is due to the calcium ions, and that causes the binding site on the actin molecule to then become exposed, which will allow then the myosin head to attach to the actin molecule. And then the next stage, stage number 3, there is obviously binding. So the myosin head now attaches to the binding site of the actin, and we say this forms cross bridges. So make sure you're using that keyword. This is caused by the ADP molecule, which is attached to the myosin head. So the cross bridges are formed due to the ADP molecule on the myosin head. We then have something called the power stroke. So this is the bit where people get a bit confused. So imagine it's like a gun that is loaded and the trigger has been pulled back, okay, or the cock of the gun is supposed to be pulled back, okay, and it wants to go off like an elastic band that's been stretched and it's ready to just be released. And when it's released, the elastic band gets smaller, and that is called the power stroke. So what happens is the myosin head then changes its angle and it moves the actin molecule long. So those thin filaments move towards the center of the sarcomere, making the Z lines move closer together. Okay, this releases the ADP as a result. The ADP has now been released. So now, because the ADP has been released, there is now room to attach a new molecule to the myosin head. So a new ATP molecule is now attached to the myosin head, and that detaches the head from the binding site, okay, and therefore the myosin head has now become removed from the actin binding site. We then hydrolyze the ATP, breaking it down into ADP and phosphate and inorganic phosphorus, okay, and that cocks the head or reloads the head, however you want to say it, of the myosin head. And here, if there is then more calcium ions, this will cause the ATP to be hydrolyzed, and that will allow that to happen to this higher energy position. The ATP has gone through hydrolysis, that's produced an ADP molecule and released a phosphate. The ADP then does the same as it did before. It causes the head to reattach to the binding site. But whereas the head was here last time, the pointer is now, the ATP from the previous stage caused it to detach, and then it was hydrolyzed, recharging it to the cocked position. It's then left ADP, which will then, which is then here. Okay, the release of the ADP will cause that to move along and push the actin filament this way a little bit more. Okay, and that will then cause the head to, more ATP will cause the head to be released and then cocked again, and then that will go back here. So that constant switch between ATP being broken down to ADP, being released and then replaced by new ATP, causes the movement of the actin molecule along this way in towards the center of the sarcomere. Notice the myosin isn't moving. Okay, so the thin filament, the actin moves, not the myosin. Now, all of this is being driven by calcium ions. So as long as the calcium ions are there, the contraction will happen. If they aren't there, the next stage won't happen. So why does that make sense? Well, when the nerve stimulation ceases, when the motor neuron prevents the acetyl, has no more action potential, the acetylcholine isn't released, okay, across the cytoplasm, the calcium ions get reabsorbed into the endoplasmic reticulum using energy from ATP hydrolysis. That means no more calcium ions are released. That prevents the ATP hydrolysis, and that prevents the head cocking and then reattaching due to the subsequent formation of new ADP. No more calcium ions also means that no more tropomyosin means tropomyosin doesn't go for a conformational change, and therefore it will go back and it will block the binding site of the actin again, preventing the actin and the myosin from attaching and forming cross bridges. And the force, and when the molecule, the opposite muscle, because remember they work in antagonistic pairs, contracts, that the force from that will pull the actin filament back out. Okay, so that's the detail you need to be aware. Just to give you some understanding of it, where does the energy come from this? Well, the energy comes from the hydrolysis of ATP, which moves the myosin head, as we talked about, and it also causes the reabsorption of calcium ions into the endoplasmic reticulum via active transport. Now, during intense exercise, you use aerobic respiration to make ATP and also anaerobic respiration to make ATP. But there is a third energy source that for A level we do not need to know a lot about, but it is called phosphocreatine. And basically you can replenish your ATP supply for a very short amount of time by splitting the phospho and the creatine up, so splitting the phosphate from the creatine to give that phosphate then to ADP to make ATP. Now, we don't need to know a lot about this at all. You just need to be aware that the phosphorus can be released from the phosphocreatine to make creatine, and that phosphorus can then join with ADP to make ATP. Now, phosphocreatine stores do not last very long at all, but we build them up in our bodies during rest, and then they'll be there ready for an emergency amount of energy if needed when we are working.